if we break it down, precision fermentation, so we all know fermentation. If we think of, sourdough, kimchi, beer, that's taking a food and then changing the properties of that food using a microorganism. If we look at beer, it's a yeast normally, and baker's yeast for sourdough. And then the precision, maybe we should talk about the biomass fermentation as well, just at that point. So we've got traditional fermentation, we all know, beer, sourdough, kimchi, things like that. Biomass fermentation is doing the same fermentation process, except the product that you're consuming is actually the microorganism that you're growing.
Interesting. So like corn.
Exactly, corn with a Q.
Yeah.
So that they do a fermentation, and then the microorganism, then they transform into the corn plant-based meat.
Yeah, and folks may remember that regular listeners, I had Paul Shapiro on, who owns a company in America called Better Meat Co. And they're doing the same thing or similar thing to corn, I think with different microbes.
Exactly.
But producing a similar sort of product.
Yeah, exactly. So then we have precision fermentation. And in precision fermentation, we are using the microorganism of choice, and there are there are literally millions of types of microorganisms, but really we're down to quite a select list, because there's a select list that are generally recognized as safe for human consumption. Things like the baker's yeast that we use to make our sourdough, we can then use that as, we call it a cell factory. So we're using the cell as a factory to produce the product that we want. And in OG Foods' case, it's dairy protein. So we give the genetic information to make a dairy protein. We give that to a microorganism, let's say a yeast, and then we tell the yeast to make the protein for us.
So let's just take a couple steps back and walk through that.
Yeah.
So when you say you give that genetic information, is that the sort of protein DNA sequence?
Yeah. So it's the DNA sequence, and you don't have to go and take any cells from a cow. We know the amino acid sequence of all of the proteins in dairy, and from that amino acid sequence you can work backwards and reverse, I guess, transcribe that into a DNA sequence, and then it's that DNA sequence that we then synthesize by like basically doing a chemical, what's the word I'm looking for? A chemical reaction to make the DNA sequence, and then we take that DNA sequence and insert it into the genome of the microorganism.
Okay. And before, when you were talking about the casein micelle, was that to better understand the sequence?
It was better to understand the structure of the casein micelle, so that we can then, I guess, understand how dairy functions like it does. Yeah.
So you take this information essentially,
exactly,
this sequencing, and you encode it or insert it into the microbe?
Exactly. We insert it into the microbe.
Okay. So, and this is done in a kind of brewery type setup?
A very, very, very small scale test tube setup.
Okay.
So basically you can make the microorganism a little bit leaky. So it's still alive, but the cell wall that kind of protects it is a little bit leaky, and then it's able to take up the DNA sequence, and then you give it a little heat shock, and then it closes back up, and then it integrates that piece of DNA into the genome.
Okay, and at that point, so this organism, basically this information goes into it, it takes that information and it starts to produce the proteins.
Yeah, there's kind of two ways. So generally we provide a switch to turn on the production of the protein. Because this protein isn't a native protein, generally they're not very happy about making them. And so what we do is we grow enough of the cells first, and we get them to a point where we've got enough cells. So we've got hundreds of millions, billions of cellular factories ready to make our product. We then tell them, we turn the switch on and say, right, make our product now, and then they start producing the protein.